Assalamu alaikum. This is the last lecture of the molecular biology part of the course. In this lecture, we'll be talking about translation, which is basically the production of protein from messenger RNA. So you can refer to Cooper's chapter 6. Okay, so in order to synthesize protein, we need a number of components. 4 major components, and these are the ribosome, which is a large structure composed of both proteins and ribosomal RNA. In addition, we need the mRNA template itself, and we need transfer RNA, which reads the codons on the messenger RNA. Now, the ribosome is made of a large large ribosome subunit, and a small subunit as well. Okay. So basically, there is a colinearity of genes, mRNAs, and proteins. What it means is that the DNA, a specific gene, is read from the 3' end to the 5' end. Okay, and it is read in that direction, and the mRNA is synthesized in the same in a direction of 5' to 3'. So it's complementary to the coding strand. Okay, so notice right here you have G, C, A, and the complementary messenger RNA reads C, G, U, etc. So there is linearity, meaning that there is no skipping in reading the bases or the codons of the DNA, of the messenger RNA and the DNA as well. Okay. Now, in terms of protein synthesis, each messenger RNA is read as a codon, or a sequence of codons. Each codon is composed of 3 nucleotides, 3 consecutive nucleotides. So it is read as GGA as an example. That's one codon. Another codon is UAC. Each codon is translated into an amino acid. Okay. Again, there is no skipping, right? So it's not like GGA, a codon, and the other codon is AUA. No, it's one after the other. One codon, one triplet after the other in that sequence. Okay, and the protein is synthesized from the N terminus, the amino terminus, to the C terminus, or to the carboxy terminus. So there is colinearity between the 3 molecules. Now, as I said, each tRNA is inserted into the ribosome, large ribosomal subunit, and it reads codons, one triplet at a time, one codon at a time. Now, the tRNA carries the amino acid that is suitable for the codon that this tRNA reads or binds to. Okay. And these are the codons. Scientists were able to translate codons into amino acids. So you can read a codon according to this table right here, and you can translate it according to this table. So the first letter of a codon, if it is, let's say, a U, the second letter of a codon is C. The third letter of a codon is A. So you go like U. Okay, so you are in one of these 4 squares. C, so you are now in this square, and you read the third letter. So let's say it's A as well. So U, C, A, and it is translated into a serine. Okay, so let's look at another example. So let's say let's say AAU. Okay, so right here, so A, the first letter, so you are in these 4 squares. Then A, another A, so you're now in this square right here. U, so that's the third letter, and it's translated into asparagine, and so on. Now, notice something. We have three codons that do not really code for any amino acid, and these are known as stop codons. So translation stops at any one of these three codons, UAA, UGA, and UAG. You are responsible to memorize these three codons. There's a fourth one, I'm sorry, and it is AUG, and it calls for methionine. Why is that important? Because usually translation starts at AUG. Okay, so the messenger RNA at the 5' end, the first codon that is read by ribosomes is AUG, which goes for methionine. So many proteins, the first amino acid in these proteins is actually a methionine. Okay. Now notice these codons. These codons have the following features. So there are 64 possible codons, and they are read from the 5' end to the 3' end as triplets. There are 3 stop codons, and the genetic code for mitochondrial messenger RNA has few exceptions, and we don't have to know these exceptions. Now, bacteria also have some exceptions, and we don't have to know these exceptions. Otherwise, the table that I just showed you is actually universal. Now, this is like sort of like an addition to the lecture of mutations, DNA mutations. So I said before that there are different types of mutations like deletion, insertion, and so on. The most common type of mutation is a single point mutation, also known as point mutation. So basically it is one nucleotide that is changed, deleted, or inserted. Okay, and such nucleotide can cause different types of mutations or variation. So sometimes you have a silent mutation. A silent mutation means that, let's say in the DNA you have an A, and this A is changed into a T. Such change does not really change the amino acid. The amino acid stays the same. So is it a mutation? Yes, it is a mutation. If it exists in less than 1% of the population, it's a mutation. If it exists in more than 1% of the population, of any population, then it is known as a polymorphism. Okay, so this is known as a silent mutation because it changes in DNA, but it does not change in the protein sequence. Now, some mutations are known as missense mutations. Now, sense means like something like logic, okay? So a missense means bad logic. And that's basically when you change the amino acid of the protein. So right here, again, you have an A, okay? You have a C right here, and this C changes into an A. So instead of C, 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 rather you have A, C, C. If you go back to that table, A, C, C codes for a threonine. So instead of a proline, you have a threonine. Okay, so because there is a change in logic, a change in sense, it means that it's a missense mutation. Another type of mutation that can take place in DNA, which can affect proteins, is what is known as nonsense mutations, meaning that there is no sense, there is no logic. Okay. And that basically when a codon changes from an amino acid codon or an amino acid coding triplet, you instead create a stop codon. So right here, notice that TAG is transcribed into UAG, and UAG is one of the 3 stop codons. So this is known as a nonsense mutation. Now, you can have another type of mutation, and it's called a frame shift mutation. And frame shift mutations are caused by addition of a nucleotide or deletion of a nucleotide. And that changes the frame of reading messenger RNA. So there's a shift in the messenger RNA. So right here, for example, you have AGC, then GTA. AGC, and then GC. So you have the TA removed. You can have, by the way, removal of one nucleotide or two nucleotides, and that would cause a frame shift mutation. What that causes is that a shift in all of the triplets, of all of the codons. So right here, instead of, so these, the CC would be combined with the G in one frame. Okay, so it's almost like putting each codon in a frame. So right here you have AGC, then GCC. Okay, that changes every frame, every codon afterwards. So the effect of an insertion or deletion of one or two nucleotides would cause a shift in the frame of reading or a frame shift mutation, and that causes the translation of a messenger RNA into a completely different protein. Okay? Now, what will happen if you have deletion or insertion of a triplet, 3 consecutive nucleotides? It depends. If the 3 nucleotides are added outside a codon, so let's say you have AGC, and then you have an insertion of 3 nucleotides right here. So what you're doing is that you're inserting an additional amino acid, but the sequence after this amino acid would be the same. So you would have serine, then any amino acid, whatever this amino acid is, then you have valine, proline, tyrosine, and so on. If insertion or deletion of a nucleotide or two nucleotides, or if insertion or deletion of 3 nucleotides occurs inside a codon, then that would also cause a frame shift mutation. So basically, it really affects frame shift mutations are really significant in terms of changing the protein. Well, how about nonsense mutation? Are they significant? Well, it depends. If the insertion of a stop codon occurs at the end, usually, if it occurs at the end of a protein sequence, then there should be little effect. Okay, but if it exists or created in the beginning of a protein, that then you will have what is known as a truncated protein. That is a cut short protein, a smaller protein, and only the first part of the protein would exist. So that would cause, again, as if some, it depends on the gene, depends on the protein, that would cause huge effect on cells. Okay. Well, how about a missense mutation? Is it significant? Well, it depends. Sometimes it is, sometimes it's not. It depends on the protein. So if you remember, like sickle cell anemia, you have a change of an amino acid from glutamate to valine. That changes the structure of the whole protein as a whole. Just a single change in one amino acid, that changes the structure of the whole protein, and that would cause a significant disease. So it depends on the type of mutation that occurs. Sometimes if it occurs, if it changes a hydrophobic amino acid to a hydrophobic amino acid, that would not cause much change. But if it occurs in a functional part of a protein, then yes, it can be significant. But there's agreement that overall frame shift mutations can cause significant effect. So let's go back to the components of translation machinery. You have tRNA, and tRNA molecules are short, single-stranded RNA molecules. They are about 80 bases long. They are linked to amino acids at the 3' end of the tRNA. Okay, and when the tRNA is connected to an amino acid, then it is known as a charged or an activated tRNA molecule. And there is a consensus sequence in the tRNA at the 3' end, and it is CCA. Okay, so all tRNA molecules have CCA, and the amino acid is linked to the A. Now, notice that tRNA molecules contain stem loop structures because there are complementary sequences within the tRNA molecule. And overall, it looks like a clover leaf. Okay, so it looks something like this. Now, what is important or a feature of tRNA molecules is that sometimes it contains abnormal bases. It does not contain a thymine, rather sometimes they contain the amino acid, the nucleotide inosine monophosphate. Okay, so sometimes you have inosine, which is an abnormal, not abnormal, a different type of nucleotide. The important part of tRNA is this here that is known as the anticodon. This is the part that recognizes the codon, and that's why it's known as an anticodon. Okay. So what happens is that when you have interaction between the tRNA and the mRNA, notice that this interaction, this hydrogen bonding between the codon and the anticodon is anti-parallel. So the mRNA goes from 5' to 3', the tRNA goes in the anti-parallel direction 5' to 3'. Okay? Translation is really quite accurate overall. And there are two things or two aspects that determine accuracy or that specify accuracy of translation. The first one is that you have an accurate association of amino acid to a tRNA by enzymes. So it is an enzymatic reactions, and enzymatic reactions overall are really specific. And there are synthetases for every amino acid. Okay, so they are quite accurate. The other thing is that there should be a match between the tRNA anticodon and the mRNA codon. But if you look back at the table that I just showed you, there is some sort of flexibility in the third codon, meaning that in some amino acids can be coded by 4 codons that differ only in the third nucleotide of the codon. Okay, so the ability of, or this flexibility in tRNA binding to messenger RNA is known as the wobble base pairing. Okay, meaning that there is sort of like a flexible interaction or binding pairing between the third nucleotide of a codon with the anticodon. Okay, this flexibility, by the way, and the fact that an amino acid can be translated the same even though the third nucleotide of a codon is different, provides sort of like a buffer against mutations. So changing the third nucleotide of a codon may not cause a serious mutation. It might cause a silent mutation. But changing the first or third or second nucleotide of a codon would cause a change in the amino acid. It would cause a missense mutation overall. But notice something, that sometimes changing the second nucleotide, if you look at the table, changing the second nucleotide of a codon may change the amino acid, yes, but sometimes it is within the same category. That is, it would change a hydrophobic amino acid into a hydrophobic amino acid and so on. Okay, so the reason behind the wobble-based pairing is that there is sort of like an angle, okay, between the anticodon and the codon. right here. So the interaction here is weak, providing sort of like flexibility of pairing between the two nucleotides. So notice something right here, like I said, changing the nucleotide, the third nucleotide would, if you look here, for example, changing the third nucleotide would not change the amino acid right here. Same thing right there. Okay. Now changing the sometimes changing the third nucleotide would change the amino acid, yes, but notice that these two amino acids are hydrophobic. Okay, so again, wobble-based pairing provides sort of like a buffer. Now, so that's tRNA, and that's how it interacts with messenger RNA. Let's look at ribosomes now. Ribosomes are really large structural proteins um, that have RNA molecules associated with them, and these are called ribosomal RNA molecules. There are many ribosomes within cells, like in bacteria. There are 20,000 ribosomes inside them. So, and they're one of the most abundant proteins that exist in cells. Now, sometimes in human cells or mammalian cells, you can have millions of ribosomes within these cells. What's really significant is that there is a peptidyl transferase activity within the large ribosomal subunit. So it's the large ribosomal subunit that forms the peptide bond between two amino acids. Okay? And exactly what's interesting is that the molecule that is responsible for the peptidyl transferase activity is actually the ribosomal RNA molecule. So RNA molecules can function as enzymes, and this is one case. Another case, by the way, whereby ribosomal RNA molecules can function as enzymes is in the process of RNA splicing, that is, removal of introns and connecting exons to each other. So ribosomes in both prokaryotes and eukaryotes are composed of large ribosomal subunit and small ribosomal subunit. This is the weight of these subunits. You don't have to memorize these. Now, the large ribosomal subunit is composed of 28S rRNA, 5.8S rRNA, and 5S rRNA. The small ribosomal subunit is mainly composed of one rRNA molecule, which is the 18S rRNA. That's in human cells. Okay. So what is S? S stands for Svedric, and it is a unit for measuring the mass of a molecule, taking into consideration its shape. Now, very briefly, rRNA molecules in human cells are produced from special genes as using RNA polymerase 1. We already mentioned that in transcription. Now, usually they are produced as a primary transcript, primary RNA molecule, and it goes post-transcriptional cleavages or modifications, whereby you have production of single ribosomal RNA molecules, as shown in here. Okay. So, as I said, the 18S rRNA associates with the small ribosomal subunit, and you have three ribosomal subunits. You have two ribosomal subunits that are connected to the large ribosomal subunit. Well, how about the third ribosomal RNA, the 5S rRNA? It is produced from a different gene transcribed by polymerase, RNA polymerase 3. Okay. Mentioned as well. So again, there are the large ribosomal subunit and the small ribosomal subunit, and what happens is that they're connected to each other, and they can be connected to the messenger RNA as well as the transfer RNA, and that's how messenger RNA is read. Now, if we look at the large ribosomal subunit, we would notice that it's almost like it has 3 rooms or chambers, and that comes to mind. Chamber of Secrets, Harry Potter. So you have like 3 chambers, okay, and they are given or spaces, and they are given the designations of A, P, and E. A stands for amino acid tRNA binding site. P is the location where you have the peptidyl tRNA binding the peptidyl tRNA binding site, or that's where the peptide bond is formed. And E stands for exit. So when you have a tRNA that is not charged, that is, it's not associated with any amino acid, this tRNA enters this room and then it exits. Okay? What happens is the following. You have a messenger RNA that is sort of like trapped between the 2 ribosomal subunits. You have the 3 chambers right here. You have a tRNA, 2 tRNA molecules standing next to each other, each one in its room. You have this binding between the tRNA, each tRNA with its codon. Okay, so you have an AUG right here. If I tell you what is the sequence of the anticodon, you would not say UAC. Rather, you should say CAU. Why? Because here's the 5' end, and this is the 3' end. So you read it again from 5' to 3'. Again, notice that this binding is anti-parallel as well. So you have these 2 tRNA molecules that are charged sitting next to each other. What happens is that you have the formation of a peptide bond between these 2 amino acids. Okay, and the direction of synthesis is going to the right. Now, just like replication and transcription, we can divide translation into 3 stages. You have initiation, elongation, and termination. Okay. So initiation starts with binding of the messenger RNA with the small ribosomal subunit. That's the first thing that happens. Okay, so you have binding of, remember, the first nucleotide, the first codon is AUG. So right here, AUG. So you have interaction with the anticodon, CAU, anti-parallel, remember. Okay, and that leads, once you have binding of this tRNA that carries methionine, that leads to engagement or binding of the large ribosomal subunit. Now you have this translation initiation complex formed. Okay. Now, so does AUG exist at the 5' end of a messenger RNA? No, not necessarily. What happens is that you have few nucleotides that precede, that come before the AUG start codon. And this region is known as the untranslated region. It's the 5' untranslated region. Now, does translation, or is translation terminated at the 3' end, at the exact last codon at the 3' end? Again, no, it does not. Remember that you have termination of transcription at the poly A site, AAU, AAA, and then you have CA right here, right? And then you have cleavage and addition of the poly A tail. So right here you can have the stop codon for translation. And this is followed by another untranslated region, and this is known as the 3' untranslated region. I do not want you to confuse terms with each other. So right here, this is the transcription start site in the DNA. It starts with whatever nucleotide, but then translation starts later on, or at a sequence that comes after the 5' end. Now here you have the stop codon for translation, and it's not the same as the termination sequence for transcription. So do not get things confused with each other. For translation, you have a start and stop codons. For transcription, you have a transcription start site, and you also have a termination sequence. Okay? So remember that bacterial messenger RNA is polycistronic. Well, so you have different parts of messenger RNA having that can produce different polypeptides. Well, each one of them has a AUG right here. So here you have AUG, right here you have AUG, and right there you have another AUG. Okay, and for protein A, or for the part that produces protein A, you have a stop codon right here, you have another stop codon right there, and you have a third stop codon in here. Remember, we also have a 5' untranslated region and a 3' untranslated region. Eukaryotic messenger RNA is monocistronic, so you have a mature messenger RNA that is spliced, it's capped, it's polyadenylated. It also has a 5' untranslated region and a 3' untranslated region, but you have one start codon and one stop codon right here. So how do ribosomes know where they should start translation in a polycistronic messenger RNA? Well, there's something known as the Shine-Dalgarno sequence. And this Shine-Dalgarno sequence is complementary to a sequence that exists in the of the ribosomal RNA of the small ribosomal subunit. It's 16S rRNA in bacteria, and it is 18S rRNA in human small ribosomal subunit. So this interaction binding between the Shine-Dalgarno sequence of messenger RNA and the ribosomal RNA would stabilize the interaction between the messenger RNA with the small ribosomal subunit. Now notice something here, that the AUG that comes right after Shine-Dalgarno sequence is the first codon that is read. Notice that there are nucleotides that are not translated right here, okay, including the Shine-Dalgarno sequence. And these are part of the untranslated region, the 5' untranslated region. So whenever you have a polycistronic messenger RNA, you have several Shine-Dalgarno sequences. You have one right here, you have another right there, you have a third in here, and translation starts at the AUG that comes right after the Shine-Dalgarno sequence. And for each one of them, there is a stop codon as well at the end. Well, how about eukaryotic cells? Do they have a Shine-Dalgarno sequence? Well, there's sort of like indication that it may or may not. It depends on the messenger RNA, but what's important is the cap itself. The small ribosomal subunit recognizes the cap. Remember how I said one of the functions of a cap in messenger RNA is to help in translation? Well, this is it. Okay, so the small ribosomal subunit recognizes the cap. Now it knows that this is the 5' end of the messenger RNA, and it looks for the AUG that comes right afterwards. So this is the untranslated region of the messenger RNA. Okay. So there is something actually in eukaryotic messenger RNA molecules for some genes, not all of them, that is similar to the bacterial Shine-Dalgarno sequence, and it is known as the internal ribosome entry site. So this sequence right here exists in the 5' end of the messenger RNA, and it is followed by an AUG. Now, this sequence right here can be recognized by initiation factors, and one of them is initiation factor 4G. Or it can be recognized by the complex of tRNA and small ribosomal subunit. So you have this interaction between all of these components. Okay. Now, why is that important? Well, again, remember when I said that the poly A tail of messenger RNA is important for translation? Even though this poly A tail exists at the 3' end of the messenger RNA, well, this drawing explains why this is the case, why the poly A tail is important. So what happens here is when you have this interaction between the small ribosomal subunit and the complex of proteins, you have interaction between the initiation factor 4G that I just told you about with the poly A tail binding protein, which recognizes the poly A tail. So once you have this interaction right here, that would facilitate the association of the large ribosomal subunit and the initiation of translation at the AUG that comes right after this interaction. Okay. Now, in bacteria, the first codon, as I said, it's the same as in human genome, by the way, is AUG, and it goes for a methionine, and methionine has only this codon right here. Now, the thing is, in most bacteria, the initiating tRNA does not carry a methionine. It carries a modified methionine known as the formyl methionine. So for many bacteria, the first amino acid of their proteins is a formyl methionine. So this is one way by which you can differentiate between prokaryotic cells and eukaryotic cells. Okay. Then you have then you start after formation of this complex of messenger RNA, tRNA, and ribosomes. Now you have synthesis starting. So you have the elongation. Again, just like any other stage, we can divide or process. We can divide it into 3 stages: initiation, elongation, and termination. So initiation again involves the interaction between the messenger RNA with the small ribosomal subunit and all of these proteins right here. Now you have this interaction between all proteins, and I emphasize on one of them, which is the initiation factor 4G that interacts with a poly A tail binding protein. That would bring in the small ribosomal subunit, and then the big one, the large ribosomal subunit would come in, form a complex, and release of all of these proteins. Okay, and you have the first tRNA sitting in the P chamber, the one in the middle of the large ribosomal subunit. Now, initiation factors are needed, including initiation factor 2 and initiation factor 4F. So these are important initiation factors. So one important protein is known as the initiation factor 2. What does E stand for, by the way? It stands for eukaryotic, to differentiate between initiation factor 2 in prokaryotic cells and eukaryotic initiation factor 2. Now, initiation factor 2 is important for bringing the initiating tRNA, which is the methionine carrying tRNA, to the messenger RNA. Okay. Now, notice that this protein is regulated, and it's regulated by GTP. Now, what happens is that after you have formation of a complex between the messenger RNA and the small ribosomal subunit, GTP is hydrolyzed to GDP. Now this protein is not active anymore. It has to be activated. And it's activated by exchanging GDP by GTP. So GTP comes in, it kicks out GDP. And then you have an active initiation factor 2 that can interact with the methionine carrying tRNA, the initiating tRNA, and it can start another round of translation. Notice that proteins in general are regulated by GTP. Okay. So you have then elongation. Elongation involves the peptide formation between two amino acids. So right here you have a tRNA that carries several of these amino acids after the formation of peptide bond, and that's how it's formed. You have now this tRNA is sitting in the P chamber. Okay, you have an incoming tRNA that has an anticodon that is complementary to the codon, and it sits in the A chamber. Now what happens then is you have the peptide bond formation. Now that these two amino acids are close to each other, you have formation of a peptide bond. So it's almost like this peptide jumps over this amino acid. Now the tRNA carries all of these amino acids, this peptide right here. And the tRNA that is sitting in the P chamber is now empty. Now there is translocation then of the ribosome, and that requires GTP, and you have elongation factor 2 that is important for translocation. Also requires hydrolysis of GTP to GDP. It requires energy. Now you have the empty tRNA sitting in the E chamber, the exit chamber, and you have the other tRNA that carries the peptide sitting in the P chamber. So this tRNA, the empty tRNA, leaves the ribosome, and now this chamber is empty. So you have the elongation factor 1. It brings in the other tRNA to the large ribosomal subunit, and it goes on and on until you hit the stop codon. So that's how it looks like. So I have an incoming tRNA. It has an anticodon that is complementary to the codon. Okay, energy is needed. Okay, initiation factor 1 is important for bringing the tRNA. Now you have these two sitting next to each other. Peptide bond formation, and notice the empty tRNA molecule right here, and then you have transcription of the ribosome as a whole, movement of the empty tRNA into the E chamber, then it leaves. Now this here tRNA is sitting in the P chamber. This is empty, allowing for entry of another tRNA molecule. Okay, so it goes on and on and on like that. Now notice that this amino acid, by the way, is at the N terminus. Okay, and this this amino acid is at the N terminus, and this amino acid now is at the C terminus. It goes on and on like this. I have this formation right here. That's how it works, and it's the ribosome that moves. Okay, so the peptide is elongated, okay, from the N terminus to the C terminus. We talked about that in biochemistry. Until the ribosome hits the stop codon. So now the stop codon is in the A chamber right here, and there is no tRNA that has an anticodon that is complementary to a stop codon. Instead, you have a protein known as a release factor, and this protein sits in the A chamber and dissociates the whole complex. So you have the polypeptide released from the mRNA large ribosomal subunit and the small ribosomal subunit. Okay. Now, there is a difference between prokaryotic cells and eukaryotic cells, and that is in prokaryotic cells, there is coupling between translation and transcription in space and time. So meaning that they can occur at the same place at the same time. The reason is that you have the messenger RNA coming out, and it comes out from the 5' end to the 3' end, right? So now the 5' end can be read by ribosomes and tRNA and so on. So you can have transcription happening at the same time as translation. But this is not true for eukaryotic cells. Rather, what happens is that they are separate in space and time. So you have synthesis of the messenger RNA 5' to 3', and then you have translation happening from 5' to 3'. Okay? So why is it that they are not coupled in space and time in eukaryotic cells? For two reasons. The first reason is that there is a nucleus that separates ribosomes from where messenger RNA is synthesized. That's one. The other thing is that translation cannot start until the mRNA is actually fully processed. So that is meaning that it is capped, spliced, and polyadenylated. So as a result, you have a structure that looks like this. This structure is known as polyribosomes. Okay, so and that exists in both prokaryotic and eukaryotic cells, meaning that you can have several ribosomes reading the messenger RNA at the same time, synthesizing the same exact polypeptide. So right here, that's DNA. This here, what you cannot see, it's covered by these black dots, and these black dots are ribosomes. So these ribosomes are reading the same messenger RNA that is coming out. Okay, but what's not shown in here as well is the polypeptide. Okay, so you look here and I ask you, where is the beginning of the gene? And you should say, it's right here. This is where the mRNA is at its shortest length. And transcription goes in this direction. Now the question is, where is the beginning? Here you have a messenger RNA coming out. Where is the beginning of the messenger RNA? Where is the 5' end of the messenger RNA? And you should say it's right here. So it's coming out 5' to 3'. Okay, you see here? Okay, so you have the messenger RNA coming out 5' to 3'. Now if I ask you, where is the longest protein or polypeptide? I'm sorry, polypeptide, not protein. Is it at this point right here coming out of ribosome, or at this point right here coming out of ribosome? You should say it's here. Here you have the longest polypeptide. Okay, why? Because this is the first one that has started reading this messenger RNAs, and it's waiting for more messenger RNA to come out of the RNA polymerase. I hope this is clear, so you can look at it this way. So right here, the one that is closer to the 3' end is the one that has the longest polypeptide. Okay. Now in eukaryotic cells, there are polysomes as well. Now, you don't have this coupling between transcription and translation, but from the same messenger RNA, you can have multiple ribosomes sitting on the messenger RNA producing the same polypeptide. Okay, so you have structure that looks like this as well. Now, this is really an efficient process. Okay, it's saving time. So whether it is the polysomes in bacteria, where you have coupled transcription translation happening at the same time at the same place, or even polysomes only in eukaryotic cells having multiple translational processes going on at the same time from the messenger RNA. Now, let's connect this to recombinant DNA technology. Recombinant DNA technology is basically forming a recombinant DNA molecule, that is, two DNA molecules from different sources connected to each other. That's a recombinant DNA. Now we can take advantage of recombinant DNA technology by inserting a gene in plasmids, for example, and this plasmid contains a promoter, so you can have transcription of this gene in bacteria. And then you can have translation as well in bacteria as well. So we can express proteins in cells. And that requires a special plasmid right here known as an expression vector. So this plasmid contains, just like any other cloning plasmid, it contains an origin of replication so that the plasmid is replicated independently of the chromosome. It has an antibiotic resistant gene, and it has a cloning site as well. Now this cloning site is preceded by a promoter, and the promoter that is used can be an efficient promoter that works really well in bacterial cells. It should have a Shine-Dalgarno sequence right here, okay? And then you have the cloning site, the place where you can cut the plasmid with a restriction in the nucleus, and you can insert a gene right here. Okay, so this is doing Western blot. If you remember Western blot in biochemistry, taking all proteins from cells or a sample and separating the proteins based on size in an SDS page or SDS polyacrylamide gel. Okay, here you have bacterial cells with no expression vector, and here you have the same bacterial cell, but these cells have the vector, and this is the protein that they produce. This is the recombinant protein. Okay, now, so in addition to the cloning site, or the in comparison with the cloning vector, expression vectors must have a promoter, they must have a Shine-Dalgarno sequence, and they should also have a transcription termination sequence as well. Okay, now we have taken advantage of bacteria to synthesize many of the proteins that we use in the clinic, including insulin, growth factor, plasminogen activator for blood clotting, erythropoietin, and so on. So this has been really useful. Except that sometimes this doesn't work for all proteins, because in some human proteins have disulfide bonds, and no such thing exists in bacterial cells. Some human proteins are modified by glycosylation, addition of sugars, and there is no glycosylated protein in bacterial cells. Some proteins are not folded properly in bacteria. They require chaperones, for example, and there are no chaperones in bacteria. And some proteins are recognized as foreign, and they are degraded in bacteria. So sometimes we can use yeast, which is a single-cell eukaryotic system, to synthesize human proteins, because they also have the molecular machinery of eukaryotic cells, like human cells, and at the same time, they are single-cell organisms. They grow fast, and they're easy to manipulate, just like bacteria. So that's basically what we do. We open up the plasmid, insert a gene, and you do have a promoter. Okay, so same idea. Now we can also do protein tagging, okay, or we can create protein hybrids as well using recombinant DNA technology. What we mean by tagging is like you have a tag associated with a protein, just like your t-shirt. It has a tag, it has a label that gives, it's like something small, or it can be big, associated with an object, like here in molecular biology. So the idea is that we have a tag that is part of a plasmid, and we can insert the gene inside this plasmid. So when the gene is expressed, and a protein is produced, it is produced with the tag that I've added. And types of tags, and these tags, tagged proteins, or the tags themselves, can be used to detect proteins, or they can be used for purification. So there are different tags right here. One of the most common ones is a polyhis tag, so it's basically 6 histidine residues. They can be added at the 5' end of genes, messenger RNAs, or the N terminus of proteins, and we can use them for detection, or we can use them for purification. Okay, sometimes we can associate large proteins with proteins. Okay, so that's the his tag. It's used for purification. Notice these are bacterial cells having the expression vector. This is the protein of interest right here. After purification, we can really purify it 100%. So there's only one protein, which is our protein. Now we can add large proteins like the glutathione S transferase to a protein that's a recombinant protein, and we can use it also for purification as well as for detection. Now we can take advantage of domains, of proteins having domains. If you remember what a domain is basically a 3-dimensional structure of a protein. It's a super secondary structure. It's composed of multiple secondary structures, and it folds independently of the rest of the protein, right? And it maintains its function. So here we have two proteins. One of them is our protein of interest, and we can associate with it a green fluorescent protein. So this protein was isolated from a gene, isolated from jellyfish, and this protein fluoresces green. Okay, so we can connect these two proteins together. This would fold independently of our protein of interest. Now this protein would fluoresce, would give a fluorescence. Okay, we can express this protein within cells with other proteins, so you can see how they are. We can associate it with actin, for example. So the actin filaments would fluoresce. This is a whole cell expressing the green fluorescent protein. Here we have the green fluorescent protein only in mitochondria, so you can see mitochondrial, the mitochondria and their structure. We can create all of these different types of organisms in here. Okay, and cells as well. So let's talk about regulation of translation now. So basically, remember how I said we have the initiation factor 2 that is important for bringing the first tRNA, initiating tRNA carrying the methionine to the small ribosomal subunit, and then you have the large ribosomal subunit coming into the complex, forming, you know, the translation initiation complex. And I said that the initiation factor 2 is hydrolyzed when this happens, and this must be exchanged, okay, in order for the initiation factor to start another round of translation. Now this happens when there is adequate growth factor, or when cells are happy. Now what happens if there is starvation is that the initiation factor 2 is phosphorylated, okay? And when it is phosphorylated, this GDP cannot be exchanged for GTP. So it's not regenerated, so it does not start another round of translation. Okay? So that basically stops every process of translation altogether, because there's no time to waste resources for protein synthesis, and cells do not grow. So here, that's global regulation of translation. Now we can also we can do this, by the way, with protein synthesis in erythrocytes, okay, immature erythrocytes or reticulocytes. Now these cells, erythrocytes, must contain a lot of hemoglobin, and it would be a waste to synthesize globin protein if there is not enough heme. So what happens is that globin synthesis stops altogether, totally, by regulating initiation factor 2. What happens this way. If there's adequate heme, what happens is that GDP is exchanged for GTP, and you can have translation going on. But if there's no adequate heme, initiation factor 2 is phosphorylated, so GDP is stuck, and you don't have translation anymore, so there's no synthesis of any protein, mainly globin, since there isn't enough heme. Remember how we talked about the regulation of ferritin and transferrin receptor, and we said that we have the iron response element binding protein, binding to the iron response element, and this protein, when it's bound, it stops translation. But if there's iron, iron would bind to this protein, releasing it from the iron response element. Now you have the formation of the translation initiation complex. You have the large ribosomal coming in, okay, and you have the small ribosomal subunit coming in, forming the translation initiation complex. So you can go back to that lecture. Now, another level of regulation is by microRNA molecules. These microRNA molecules, without going into details, what they do is that they can bind to, they can be complementary to part of the messenger RNA. So you have this binding of the microRNA to messenger RNA, and that would lead to mRNA degradation or block of translation. So when you have binding of the messenger RNA, specifically at the 3' end, at the 3' untranslated region of messenger RNA, you do not have this connection between the poly A tail with the proteins at the cap. Remember how I said that this interaction is important for translation to start? Well, the microRNA would prevent the formation of this interaction between the two, the poly A tail with the proteins at the cap. Okay, so microRNA molecules are quite important. Finally, we will end with protein folding. This is another level of regulation. So basically, proteins do not last forever, and they are degraded, and there are different mechanisms of protein degradation. One of the most famous, one of the most common ones is by proteosomes, which are large proteins. Basically what happens is that protein is ubiquitinated, meaning that you have addition of these small molecules, lipid molecules, to proteins, and that labels a protein for degradation. So proteosomes recognize these proteins, and you have degradation of proteins. So that's another level. So the idea is that here you have different levels of regulation, and we have talked about throughout the course about these different levels. You have regulation of transcription, you have regulation of RNA processing, an alternative, splicing, capping, polyadenylated, and so on. Regulation of RNA transport, so some RNA molecules are not transported out of the nucleus. mRNA stability, we talked about that. Remember, when we talked about translation, we also talked about the euchromatin, heterochromatin, the compaction of the chromatin structure, epigenetics basically. We talked about translation and regulation of translation. You also have post-translation modification of proteins, like glycosylation, phosphorylation, and so on. In biochemistry, we talked about protein activity, like enzyme activity, okay, and how this is regulated as well. And finally, protein degradation. All of these different processes are controlled and well regulated, and that's what makes us human beings. So this is molecular biology in a nutshell. Allah ma'akum.